So the first speaker is Amy Brunton-Martin, and Amy is a postdoc who is working on a Marsden-funded grant to look at the hottest topic in biology at the moment, which is truffle-like fungi. And so I think I might just hand it over to you, Amy, and I'm going to mute myself.
I'm going to unmute myself. Okay. I'll just start. All right, so hi everyone. As David said, I'm Amy. I'm a postdoc at Manaki Whenua Land Care Research. Hopefully you can all see my screen. I'm assuming you can. And can you see my little laser pointer? Yep. Excellent. So yeah, I'm going to talk to you a little bit about some of the work I've been doing there for the last year. I'll just start off saying I did my PhD on sexually deceptive orchids in Australia and New Zealand. So fungi research is new to me, but for the last year and a half, I've been at Manaki Whenua, looking at New Zealand's native truffle-like fungi. So first up, a quiz, which I now realize is going to fall down a little bit because I don't have any microphones, so I'll just imagine, for all of you people here for the fungal foray. So I'm going to ask you to spot the difference. Can you? I'm going to give you like a moment. And now I know you're all on the edge of your seats. You want to know the answer because you're having such a hard time spotting the difference. But some of these are fruit, believe it or not, and some are truffle-like fungi. More specifically, these are berries and truffle-like fungi that are native to Patagonia. So they resemble, the fungi resembles berries in terms of their shape, size, and color. Now, if you had maybe even a little bit of trouble detecting a difference, do you think other potential dispersers would? Well, certainly this bird couldn't tell the difference or didn't care to tell the difference. This is the black-throated huit-huit. And this bird, whose name I might butcher, and I'm sorry in advance, the Chukau tapaculo, also didn't, because both of these bird species, as well as some others, regularly consume both these berries and their truffles across Patagonia. They've been observed eating them, and also their DNA is found regularly in their feces. So bearing all this in mind, I'd like to now present you with another spot the difference. Maybe a harder one, I don't know. So can you spot the difference here? And this time, actually, it might be easier because you've got your eye in now. So here we have some fruit, and here we have some fungi, which are fungus, which maybe you have spotted in your foray for the last couple of days, maybe not. It's reasonably common. This is the scarlet pouch truffle, or Loradiomyces erythrocephalus. And it's native to New Zealand. It's brightly colored and emerges from the forest floor. And you might mistake it for a fruit. But everywhere else in the world, apart from Patagonia maybe, truffles and truffle-like fungi are known for being buried, dull-colored, and dispersed by mammals, mostly using scents to attract their dispersers. But here in New Zealand, we're thought to have a kind of wide array of brightly colored truffle-like fungi, and it's evolved many times. And they all emerge from the forest floor. They have no obvious mechanism for direct dispersal into their environment because their spores are fully enclosed. But they have proliferated across New Zealand. So how might that have happened? So Ross Beaver proposed that the unusual traits that we see within the New Zealand truffle-like fungi, so the flora, fauna, and funga, could reflect adaptations for dispersal by birds and reptiles rather than mammals. The endemic terrestrial fauna of New Zealand was, after all, predominantly these groups. We only had 2 species of bats to represent mammals. And this is thought to explain why, in theory, our truffle-like fungi are so much more colorful than everywhere else in the world. Convergent evolution between fungi and plant fruits to look like this food source, something that would attract these dispersers to eat them and disperse their spores. They've all converged upon the same kind of key traits, much like what has kind of recently been observed in Patagonia. I mean, that work was done last year. But wait, this has been said that New Zealand's truffle-like fungi are much more colourful than everywhere else in the world and repeated in the literature a lot, but has never actually been quantified. So I thought, as my first point of call for my postdoc, this is something that needed looking at. So how to do this? There's no way that I can actually sample all the truffle-like fungi in the world, at least in the course of a postdoc, and quantify their colors and shapes myself. But that's where you all come in. So as was talked about on Monday, I used something called the GBIF, Global Biodiversity Information Facility, to get an overview of what truffle-like fungi are like globally. So I can use things that you all put up on platforms like iNaturalist, and maybe you contributed to in the last couple of days, to look at this, to get an assessment of this. And it is especially useful when you're stuck in various lockdowns for the first year and a half of your postdoc. I think I've spent approximately 10 months in one lockdown or another. It means that there's still something I can do, even though I can't really get out into the field. But what I did was kind of collated together a list of species of different truffle-like fungi across the globe. I did this using literature searches as well as guidebooks. I made sure that the species list included those that are only species for which we have no record of a kind of active spore dispersal mechanism. So this would then exclude things that look like they could be truffle-like fungi because they're nice and closed up, but they aren't because they'll release their spores into the wild, like a puffball. And then I took my list of species and I searched for these species across the world using the GBIF database. And I had many records from that, about 90,000, maybe 80-something thousand. And then because there's a not negligible amount of bias in this sort of data, you get problems with bias with citizen science or museum collection data, because it's just a matter of people go out and sample the same places over and over again, so you get more things. So to avoid this bias as much as possible, you have to clean it. So that meant that I went through and I removed records, for instance, that were close to capitals or to research hubs. And I did something called spatial thinning based on how quickly by road you can get to a certain location. So the easier a location is to reach, you might think there might be more fungi found there. So I did some spatial thinning to randomly remove some observations from there to make it a bit more even. I also removed things like observations from before the 1950s, because often those can be unreliable in terms of their GPS coordinates, among some other things. And once I have this list of observations, I ended up with about 30,000 records. So it cut down a lot of observations. And I'm still figuring out the best way to kind of manage that bias. But I went through and categorized these observations into two types of truffle-like fungi. They either have a bird syndrome or not. And how I defined a bird syndrome was that the truffle-like fungi had a diameter that's smaller than the gape size of the largest living or extinct frugivorous bird. It also has to be noted in its description as either above ground or emerging from the forest floor. And also I noted its color, and I lumped them into either being a contrastive color or a dull color, because birds in particular are known to eat contrastive things if you go off of the plant fruit literature. Whereas mammals typically go for less contrastive things that are smelly. So in the literature, I was looking for color descriptions such as blue, red, white, or purple, and orange, and these are contrastive. And if they didn't fall into that category, they were considered dull. And once I'd considered this and categorized them, I ranked the truffle-like fungi. They got a 1 if they were considered bird syndrome, and a 0 if they weren't. And then if you map it so far, going off those 30-something thousand records, if you map it across a 200-meter square grid, you can calculate something, calculate the average prevalence, which is what I did. So here, this is a 200-meter square grid across the world of truffle-like fungi. So the average prevalence is how many bird syndrome truffle-like fungi on average can you find in this 200-meter square grid. And the more orange, the closer to 1 it is, so the more bird syndrome truffle-like fungi you have, the more green, the less you have. And you can see that here in New Zealand, it's quite a bit more orange than elsewhere in the world. But it's important to also note there are some other places, for instance, here in South America, where we have evidence of bird dispersal of truffle-like fungi. We also have these brightly colored, we also have a higher prevalence of this bird syndrome truffle-like fungi. And then I went through and tested for a statistical difference here, accounting for differences in sample sizes between regions. So some areas like Africa have quite low in terms of observations. But if you account for those differences, you can find that the odds of finding a bird syndrome truffle-like fungi, it's a mouthful, significantly higher in New Zealand than anywhere else in any other region that I had data for. And notably also, the odds in South America are also higher than any other region, except for New Zealand. So to answer that first question, are they more colorful here in New Zealand? Do we have more of this kind of truffle-like fungi that seems to be targeted towards bird dispersal? At least going off of the citizen science mixed with museum collection data, it does seem, yes, that they are. But you see, what's important to also understand is that, at least in terms of the color here, I've taken many of my coloration categorizations based on human observation, which is really useful for these kind of broader studies, because I would not be able to look at color of all truffle-like fungi. But at least in terms of in New Zealand, you can take a different approach to kind of get the point of view of our dispersers and our native dispersers, because humans don't see the world the same way as other animals. So humans, for a bit of background, trichromats. So we have 3 different types of cone cells. That's these 3 here. And this graph kind of shows where they maximally absorb light. So here's the UV end of the spectrum, and here's our visual spectrum, so here from blue to red. And we maximally absorb light in this area, and this area. And in contrast, birds, tetrachromats. So they see in blue, green, red, but they also see in UV often, sometimes not. But if we're talking about convergent evolution, it's important to acknowledge the perspective of the animal that, in this case, the truffle-like fungi might be aiming for, and test whether those potential dispersers can actually see truffles against the forest floor and recognize them as a potential food source. So you would expect, if they have converged on this one strategy, that potentially one truffle-like fungi that's red has very much the same color or looks the same to a bird as a plant fruit that is red and on the forest floor. And this brings me to the second aspect of this talk, which is looking at how dispersers might see these things. And to do this, it typically involves me locating a truffle in the wild and then using a spectrometer, which is this machine here. This is a portable spectrometer, but we also have some desktop ones, to measure its color. And the thing you're measuring with this machine is its spectral reflectance. And that's what looks like this. And so these are the spectral measurements of some of New Zealand's truffle-like fungi. Interestingly, these 3 here, here, and here it's up here, reflect in the UV, at least a little bit in some cases, or a lot in other cases. But otherwise, they're pretty standard in terms of their color. Their blue one is, the Clavogasta virescens is blue. It reflects mostly in the blue end of the spectrum, the blue middle of the spectrum. And the red ones reflect in the red end here. This one is a bit more orange. And the lines are colored based on a modeled human perspective of the spectral reflectance here. I also have an array of spectral measurements from fruit, and some give off some UV here and here, and very much here, and otherwise not so much. So this is a dark colored fruit. It's not so surprising. Purple fruit comes out as purple, red is red. It's amazing. And the next thing to do with these measurements is to take them and model them into the visual system of an animal that might disperse them. But we have a problem here because in New Zealand, many of our potential dispersers are extinct or functionally extinct. For instance, we have evidence of truffle spores in ancient moa and kakapo coprolites. And if these birds are eating the truffle-like fungi, they might be mistaking them for fruit. But because moa, who are potentially a known disperser, are extinct, I can't get the particular information about their eye structure that I need. So I would have to rely on some approximations to model their vision of a truffle-like fungi. So what might be some good approximations of a moa? Well, for this example in this talk, let me introduce you to 3 related ratites as proxies for moa vision. 2 with good UV vision, an ostrich and an emu, and 1 with poor UV vision, so a rhea. And this is just to make sure I show a range of perspectives and consider kind of a range, which is particularly important when you can't actually model the living animal's eye. And the next thing to do is to see whether this moa or these birds can detect fruit or truffles on the forest floor. So if they're walking around and they look down on the ground, would they be able to see it against the forest floor? So what I do is I take the spectral measurements of the fruit and the fungi and the information about eye sensitivities, so where they absorb maximally, as well as how many cone cells there are in their eyes. And I can model in R the visual systems of these animals. You can also do it in MATLAB and Python if you want to, but I like R. And the basic output of this mathematical modeling is something called a just noticeable difference. And essentially, if the value is greater than 1, so at 1, the animal can just detect a difference, in this case between the forest floor and the fruit or fungi. Just notice a difference at 1, but only in really good lighting. At 5, they can just notice a difference in natural lighting, and any number bigger than that, it means it's more discernible and the animal is better able to detect a difference, in theory. So let's see what we get when we look at all my fruit and truffle measurements against a background, in this case, a leaf litter background, just to see whether a moa might be able to see a truffle or a fruit against the forest floor. So I've represented those thresholds here with a dotted line, meaning that it's discernible in nature, and a solid line, meaning it's discernible in ideal lighting. And what I present now is this JND, the just noticeable difference value for the fruit and the truffles, and that I showed earlier. So those ones that I had spectral reflectances for when contrasted against a leaf litter background. And what I found is that they're all above this dotted line. So that means that both fruit and truffles are quite easy to see against a leaf litter background. Interestingly, the scarlet berry truffle, so Porcotilus pila here, and here, is maybe a little bit harder to see for all 3 against leaf litter, but they can do it. And rhea overall is not so good at distinguishing everything against the forest floor, but it can still do it. And this might be because of its weaker UV vision, particularly because many of those plant fruits and truffle-like fungi did reflect in the UV. So bearing all that in mind, we can say that yes, our extinct birds could potentially see truffles against the forest floor. And whether they recognize them as a potential food source, or whether it seems like color at least has converged between truffle-like fungi and plant fruits in New Zealand, well, you will just have to wait and see, because we have a really fantastic group of people working on this project. So we are analyzing coprolites and looking at isotopes in bones. We have trail cameras set up on truffle-like fungi. Kyung-Soo Lee, an honors student, actually he's just finished his honors and he's going to start a PhD on the same project, is looking at the sensory capabilities of our native birds, both living and extinct, and linking it to their ecology and their potential as dispersers. Gabrielle Johns, our master's student, is taking many more spectral measurements and much more, which maybe she'll be talking about next year, if I can convince her to come along. And she's modeling what I talked about, so this convergent evolution where the plant fruits look the same as truffle-like fungi. And she is also looking at this for a variety of potential different dispersers. And with this kind of multidisciplinary approach, hopefully we can understand a bit more about fungi dispersal in New Zealand and the group as a whole. So thank you all for listening, and hopefully I will have even more results for you next Fungal Foray.
Fantastic. That's really good. Thank you very much, Amy. Gosh, what a exciting project. You've only left yourself about 1 minute for questions. But,
I know.
I'm interested to, if anyone, Pauline's clapping there, that's good. If anyone wants to ask a question, then you could raise your hand and I might be able to let you in, but otherwise you can pop them in the chat to everybody or to Amy. Oh, there's a question up from Laura. Yay. I can do this. Right, Laura, you should be able to share your video now, or your audio, or whatever you want. There we are.
Thank you. Thanks, Amy. That was very interesting. So you're looking to see whether they eat the fungi because they look like, or they might be mistaking them for fruit. Is there any benefit that the birds could get just from eating the fungi in its own right? Like, do they get nutrients from it? Could they just be choosing it because it's good?
Yeah, so I don't know. Yeah, exactly. So it's less of the idea that the fungi are tricking the birds into eating it because it looks like fruit. It's more that they've converged on the same sort of strategy, I think. At least with the truffles that are eaten by birds in Patagonia, they get a protein value from it. I don't know about the species for sure in New Zealand, but that's kind of as far as it goes. So yeah, they might be eating it because it looks tasty, but the truffle have got to kind of grab their attention in the first place anyway.